Welcome. I'm Tony Caggiano. I'm the Chief Medical Officer and Head of R&D at Cognition Therapeutics, and today we're hosting our next in a series of conversations hosted by Cognition Therapeutics with key opinion leaders and experts in the field of development in neurology, particularly focused on Alzheimer's disease, dementia Lewy body, and related disorders. Today, we have the privilege of talking with Drs. Anthony Porsteinsson and Dr. Christopher Van Dyck.
Thanks, Tony. My name is Dr. Anthony Porsteinsson, and I'm the William V. and Sheila Kohner Professor of Psychiatry, Neurology, Neuroscience, and Medicine at the University of Rochester School of Medicine. And basically, my career is focused on the care and study of people with Alzheimer's disease and related dementias.
Thank you, Tony. I'm Chris Van Dyck. I am Professor of Psychiatry, Neurology, and Neuroscience at Yale School of Medicine. I also direct the Alzheimer's Disease Research Unit. And I work primarily in clinical research in the therapeutics and neuroimaging of Alzheimer's disease.
Thank you, and thank you as well for joining us. So Cognition Therapeutics has had the pleasure of working with both Drs. Porsteinsson and Van Dyck in the past. Dr. Porsteinsson is part of our steering committee for our START trial, which is a trial of CT1812 in early Alzheimer's disease. And Dr. Van Dyck was an investigator, or the investigator, in a neuroimaging study we conducted with CT1812 and will be our lead investigator in our START trial, which will be starting soon. So Chris, we'd like to start with you. A few weeks ago, we all had the pleasure of watching you present data from the lecanemab trial. So we'd love to hear, obviously we don't need to go through all of the details again, we'd love to hear what you think the highlights of that data are and the implications.
Yeah, well, thank you, Tony. I think the highlight is really that for the first time in our field, in Alzheimer's therapeutics, we have a therapy which has unequivocally demonstrated a slowing of progression in people with early Alzheimer's disease. So, and that was to the tune of, on the primary outcome, a 27% slowing of decline, and then in several other measures of cognition and function, similar rates of decline in the order of about 25 to 35%.
So Anton, obviously you were there as well to see Chris present, and additionally we saw a presentation with gantenerumab. Is there anything you took there beyond the highlights we've just talked about, which excite you about the state of development here in Alzheimer's disease?
This was really a tour de force presentation that answered a lot of questions that, at least I came in with what I wanted to hear from lecanemab and the sponsors and the team. Chris and I will share probably our disappointment that the gantenerumab data failed to show a separation between the drug and placebo. And it seems to be that there was some maybe flaw in the modeling. Here we were using a subcutaneous drug delivery, and the reduction in beta amyloid plaque burden certainly wasn't what we expected it to be. And the clinical outcomes were softer.
Sure. So Chris, maybe you can expand a little bit upon those thoughts around removal of amyloid plaque, and in your mind, does this study and these data now clearly demonstrate the utility of going after amyloid as a target in Alzheimer's disease?
In terms of going after amyloid, I mean, some thoughts about that. Well, one thing, of course, is that there's some people who look at these data and say, aha, we've proven the amyloid hypothesis, right? I mean, the people who are most associated with the amyloid hypothesis will look at these data and say, yes, they prove that we were right. I have a broader view of that. I think Alzheimer's is a complex disease. I don't think I'm alone in that. I think it's multifactorial, and I think there are multiple potential therapeutic targets along the way. Amyloid is just one of them. I think the trial demonstrates that amyloid is not irrelevant for Alzheimer's disease, if anybody thought that. And one of the things I think we'll probably talk about is even among amyloid targets, these are amyloid lowering therapies we've been talking about. but there are other ways to go after amyloid, such as blocking, its effects, in other ways.
And Chris, can I ask you? Lecanemab is actually a unique antibody. It has maybe a different binding profile than some of the others. What do you think? Is this kind of broader binding profile and the more reach and higher affinity for oligomers and protofibrils? Do you think that matters?
So Anton, yeah, no, it's a great question. I think it probably does matter. And let me just say, kind of as an overarching thing, I'm kind of uncomfortable with some of the talk, that we hear now about, it's about if we lower amyloid, we achieve benefit, as though amyloid is all one thing, right? Of course, I think we know it's not. There are, different conformations of amyloid, just as you're alluding to. And when we do the PET scans, all we're looking at, right, is this fibrillar amyloid, in plaques. So it's not clear that, at least in my view, that it's necessary or sufficient to clear plaques from the brain to have benefit. That could be a surrogate for clearing other aggregated species, right? Like if you may be clearing plaques, but also clearing oligomers and protofibrils, that you mentioned, and it's the latter that's really more important.
So speaking of those other toxic amyloid species, such as oligomers, Chris, maybe you can mention kind of things that you see as promising avenues for addressing those species as well as the larger plaque, which we so effectively remove with antibodies.
Right. So exactly. Thank you, Tony. Yeah, I was sort of touching on that with an eye to expanding a little bit. Sure. But right, even among anti-amyloid therapies, right, a multi-pronged approach, so clearing amyloid, clearing at least initially, oligomers, protofibrils, may be beneficial, but we also have the possibility of interfering with the toxicity of those oligomers that are there. And that is where, a therapeutic like, CT1812 comes in, so CT1812, and we'll all be, engaged in this trial that you alluded to before the START trial, with the ACTC. This is a therapeutic that, binds the sigma-2 receptor, and in so doing interferes with oligomer binding to putative oligomer receptors on the surface of neurons, and thus potentially blocking toxicity. So that potentially could be additive or even synergistic with a therapeutic that's also clearing oligomers. And so that's an example of where I think there's still potentially more to gain, even in the anti-amyloid realm, with a kind of a complementary therapy to one like lecanemab.
So Anton, speaking of those trials, what are your thoughts on combination therapies, both in research trials as well as one day, hopefully, in an approved setting where we're treating patients?
Yeah, Tony, I think there is no question that there's unlikely to be a singular solution to Alzheimer's disease, even if we move to the preclinical stage. Alzheimer's disease, there are amyloid issues, so amyloid dysregulation, and the oligomers and the protofibrils and the plaques. There is tau, particularly p-tau. There's neuroinflammation. There's oxidative stress. All of this creates an incredible strain on the brain cells and leads to accelerated death. There's probably more than one way to approach each one. So we've talked about amyloid. Well, there is the plaque-busting antibodies that target the N-terminus. There are some antibodies that target more soluble amyloid. There is CT1812 that prevents the synaptic damage. There are others like ALZ801 that decreases the fibrillization of amyloid. There are active vaccines against amyloid, and that's just the amyloid space. But we can't be happy with where we're at. We have to basically carve a path forward.
Yeah, absolutely. And obviously we're excited about trying to demonstrate the utility of CT1812 on its own, but now even more excited about the potential, like you mentioned earlier, of possibly synergistic effects of removing amyloid and then with our drug attempting to block the toxic effects of remaining oligomers. So hopefully the next few years will tell, and we'll have some positive data coming out.
And Tony, let me though add one thing, and that is that lecanemab and similar medications are not for everyone. We have to select patients. They can't have certain conditions. They can't be on certain medications. These treatments are cumbersome. So not everyone is going to even want to be on this kind of treatment. So we have to be able to offer two options. You know, there are some people that will be on a combination treatment. There are others that will not be interested or not able to be on some of the new medications. So number one, we have to find other options that may be as effective or more effective or have a different safety and tolerability profile. And we also have to be able to, add. Here we have, like Chris said previously, a 22 to almost 40% slowing of decline compared to placebo. But in both situations, patients are declining. So, we hope with combination treatments that we can do better than this.
Yeah, those are very, very important points. Well said, Anton.
Yeah, so we've obviously spoken a lot about lecanemab and CT1812, but both of you have a very unique perspective being researchers in the field of Alzheimer's, and you see a lot of things that are in development now. Using your crystal ball, what do you see in the next 5 and 10 years, and how will we be able to bring approved drugs towards individuals, and what will that treatment regimen look like?
If we try to predict, we're going to be wrong. There are going to be mechanisms that'll appear that we hadn't even considered, and we have to figure out then how to integrate them in studies. I think some of the things beyond what we've talked about, amyloid, tau, there are the studies. Well, Anton has alluded to some of the other mechanisms, but things, for example, particularly promising area, is energetics, metabolics, insulin resistance. I think therapies like that are definitely going to continue to emerge and be tested, and they can be combined very well with for example, anti-amyloid therapies if those become to some extent standard of care.
Just being in this space right now is definitely more exciting than at any other point in my career. That's absolute.
Well, we really want to thank both of you for taking the time to have this discussion with us. We want to wish you a happy holidays, and we look forward to working with you on the START trial here over the next several years with CT1812. So thanks again, and we'll see you soon.
Thank you. Thank you, Tony.
Thanks, Tony, and thanks to the team as well.